Hello everyone, welcome to biotechnotebook. In this video we are going to see in detail about cells of the immune system. All the immune cells originated from a single type of stem cell which is known as hematopoietic stem cells. Not only immune cells, all the blood cells are arises from this hematopoietic stem cells. So first let's see about what is stem cells and types of stem cells and what is hematopoietic stem cells. Let's define stem cells. Stem cells is defined by two capacities. The first one is to regenerate or self-renewing capacity. Through this self-renewal process, each stem cells can produce more number of daughter cells. Each stem cell can divide into two daughter cells, and each daughter cell can also divide into two daughter cells. Like that it can produce more number of daughter cells, and all these daughter cells in an undifferentiated state. Then your second property of stem cell is differentiation. Through this differentiation process, stem cells can differentiate into all diverse cell types. That means stem cells can differentiate into all types of cells present in an organism. So differentiation is the process whereby our stem cells become more specialized cell types. Okay, there are different types of stem cells. In that, the two main important types are embryonic stem cell and adult stem cells. Embryonic stem cells have the capacity to generate every specialized cell type in an organism. So this embryonic stem cells also known as pluripotent. Okay. Then adult stem cells have the capacity to give rise to the diverse cell types that specify a particular tissue. Okay. Adult stem cells can't differentiate into all the types of cells in an organism, but it can differentiate into cell types that specify a particular tissue. For example, hematopoietic stem cells and skin cells. Adult stem cells are also known as multipotent because it can produce many types of cells in the same tissue. So hematopoietic stem cells are example for adult stem cells because our blood has many different types of cells. All the cells came from a single cell, that is hematopoietic stem cells. So hematopoietic stem cells can differentiate into all types of blood cells. As we know that our immune cells originates from this hematopoietic stem cells. Okay, so the process by which hematopoietic stem cells differentiated into all types of blood cells is known as hematopoiesis. Okay, so hematopoiesis is the process by which hematopoietic stem cells differentiated into all types of mature blood cells. Then in blood cells we have three types. One is red blood cells, then white blood cells, then third one is platelets. Okay, red blood cells also known as erythrocytes, and function of these red blood cells are transport or carry oxygen from the lungs to the body's tissues, and it remove the carbon dioxide from the body's tissues, and it return back this carbon dioxide to the lungs to exhale from the body. Then platelets. The function of platelets is to prevent and stop bleeding by forming a blood clot. Then these platelets and red blood cells are not part of our immune system. Only these white blood cells is a part of immune system. So in this video we are going to see in detail about these white blood cells. These white blood cells are also known as leukocytes. Okay, the function of white blood cells is which protects our body from the infection. So white blood cells are the vital part of our immune system, and these white blood cells search and destroy the foreign invaders like bacteria, fungi, yeast and viruses, and also it eliminates some foreign molecules entering into the organism, and also it eliminates tumor cells and cancer cells. Next, let's see what are the cells arises from this hematopoietic stem cells. Okay, and these hematopoietic stem cells also have the two important capacity of stem cell. Okay, so it can self-renew itself. Okay, so these stem cells can divide into daughter cells. In these daughter cells, some retain their self-renewal capacity. Okay, give rise to same hematopoietic stem cells. Okay, and some daughter cells enter into differentiation process. In differentiation process, these daughter cells proliferate and differentiate into lymphoid progenitor and myeloid progenitor. Then these differentiated cells loses its self-renewing capacity. and become progressively more committed to a particular lineage. Okay, if the daughter cell differentiated into lymphoid progenitor, then it gives rise to T cell progenitor and B cell progenitor. It also gives rise to natural killer cells, then dendritic cells. Okay, then this T cell progenitor give rise to two types of cell. One is T helper cell and then TC cytotoxic cell. In T helper cell, you have different types of subsets. Later in this video, we will see in detail about the subset of T helper cell. Okay, then B cell progenitor gives rise to B cell. Myeloid progenitor cells differentiates into granulocyte monocyte progenitor, eosinophil progenitor, basophil progenitor, erythroid progenitor, then megakaryocytes. Okay, so this granulocyte monocyte progenitor gives rise to monocyte, then neutrophil. Okay, monocyte differentiated into macrophages in tissues. Okay, monocyte circulates in blood and it differentiate into macrophage in tissues. Okay, then it also differentiated into dendritic cells. Eosinophil progenitor gives rise to eosinophils. Basophil progenitor gives rise to basophil mast cells. Erythroid progenitor gives rise to erythrocyte. Okay, this erythrocyte helps in transporting oxygen and carbon dioxide. Then megakaryocytes. Okay, megakaryocytes differentiates into platelets. Okay, it has the function of preventing and stopping bleeding by forming a blood clot. Then the dendritic cells, the only cells which can arise from both lymphoid progenitor and also from myeloid progenitor. Next, let's see in detail about each cells of the immune system. First, let's start with lymphoid progenitor, that is lymphocytes. So these lymphocytes are 20% to 40% of circulating white blood cells. Okay, so in humans, approximately a trillion, that is 10 power 12 lymphocytes circulates continuously through the blood and lymph, and then it migrates into the tissue spaces and lymphoid organs. Okay, on the basis of functional and phenotypic differences, lymphocytes can be classified into three types: B lymphocytes, that is B cells; T lymphocytes, also called as T cells; then natural killer cells, that is NK cells. Then these lymphocytes are nondescript cells. That means that are difficult to distinguish morphologically. The T and B cells in particular appear identical under microscope. So T and B cells are differentiated by surface proteins and antigen specific receptors. Surface proteins are known as cluster of differentiation. These surface proteins are expressed on the cell surfaces of B cell, T cells, and there are different types of cell surface proteins. So based upon that, we can differentiate T cells, B cells. Then antigen specific receptors present on T and B cells also used to distinguish these T and B cells. So B cell express B cell receptor BCR in its surfaces, and T cell expresses T cell receptors, that is TCR, and on its surfaces. So each B cell and T cell expresses a billion type of many different types of B cell receptors. Each receptor has their own structure, so it specifically goes and binds with a specific antigen. Okay, next let's see some technical terms before seeing in detail about T lymphocytes and B lymphocytes. So first let's see what is clone. Okay, if a given lymphocyte divides to form two daughter cells, then these daughter cells are known as clone. Why? Because both these daughter cells bear antigen receptors with antigen specificities identical to each other. Okay, they both have the same antigen receptors on their surface, and that receptors have the same antigen specificities. Okay, and also which are similar to the parental cell, okay, from which they arose. And also if it divides into two daughter cells, then these cells also have the same similar properties to the parental cells. Okay, the cells formed from a single lymphocyte all known as clones. So clones mean they all have a similar properties. Then naive B cell and T cell means mature B cells and T cells are ready to encounter antigen, but they are considered naive B cell or T cell until they encounter the antigen. Until they do so, that is until they encounter the antigen. Mature B cells and T cells are known as naive B cells and naive T cells.
The next term is effector cells and memory cells. Okay, so once the mature B cell and T cells contact with antigen, it induces the naive cells, that is the mature B cells and T cells, to proliferate and differentiate into both effector cells and memory cells. Okay, so once the naive cells contact with antigen, it will proliferate and differentiate into effector cells and memory cells. Okay, effector cells and memory cells have their own specific functions. Effector cells carry out specific function to combat the pathogen, that is, it will fight with the pathogen. Effector cells are action cells, but memory cells persist in the host and it will act only upon re-challenge with the same antigen. Okay,
so these memory cells persist in the body for a long time and it gives long time protection against the same pathogen. If the same pathogen reinfect the host, these memory cells will respond very quicker and greater in magnitude.
So the first encounter with antigen is termed as primary response. Okay, if the host is encountered with an antigen, there will be effector cells produced in the host that will clear the antigen, and that response is known as primary response. Okay, and the re-encounter with the same antigen. Okay, if the antigen second time it will come and attack the host, then that is termed as secondary response. Then this secondary response is very quicker with greater magnitude. Okay, first let's see in detail about B lymphocytes. The B lymphocytes derives its letter, that is B designation, from its site of maturation. Okay, in birds these B lymphocytes are matured at birth of Fabricius, and in humans, mice and many other mammals, these B lymphocytes are matured at bone marrow. First letter of bone marrow and birth of Fabricius
denotes the letter B.
Then mature B cells are distinguished from other lymphocytes and all other cells by the synthesis and display of B cell receptor on its surface. Okay, this B cell receptor is also known as antibody molecule, that is a membrane bound immunoglobulin molecule. Okay, this antibody goes and bind with the antigen. Okay, then each B cell expresses a surface antibody with a unique specificity. Each of the approximately 1.5 to 3 into 10 power 5 molecules of surface antibody has identical binding sites for antigen. Okay, each antibody has its own specificity for the specific antigen. Then B cells can also improve their ability to bind antigen through a process known as somatic hypermutation. Okay, this process introduces mutations into B cells, thereby it can create a wide variety of antibodies. Okay, so this process helps the immune system to fight with a new foreign elements. Okay,
then there are different types of antibodies. Different types of antibodies are produced by B cells by the process known as class switching.
So we have 5 classes of antibodies. Okay, so each antibody has different function. So these classes are
IgG, IgM, IgA, IgE and IgD. Okay, next let's see what is plasma cells.
Already we have seen, once naive cells encounter an antigen, after that only it will get activated and it will proliferate and differentiate into effector cells and memory cells. Okay, so these effector B cells also known as plasma cells. Okay, so these plasma cells lose expression of surface immunoglobulin. Okay, so it will lose the surface antibodies and become highly specialized for secretion of antibody. Okay, so it loses the expression of surface immunoglobulins and it starts to secrete a particular antibody. Okay, so a single plasma cell is capable of secreting from a few hundred to more than 1,000 molecule of antibody per second. Okay, then these plasma cells do not divide and it may die within 1 or 2 weeks. Okay, the next, the immune response given by B cells, also known as humoral immunity. Okay, humoral immunity is a type of immune response that uses antibody to fight with pathogen. Okay, so these type of immune response is also known as antibody mediated immunity. This type of immune response is given by B cells. Then the immune response given by T cells are known as cell mediated immune response, which we will discuss later in this video. So immune response given by B cells are known as humoral immunity, and immune response given by T cells are known as cell mediated immunity. Next, let's see about T lymphocytes. Okay, these T lymphocytes derive their letter designation T from their site of maturation in the thymus. Okay, the first letter of thymus T denotes these T lymphocytes. Okay, like the B cells, the T cells also express a unique antigen binding receptor, which is known as T cell receptor. As we already seen that B cell has a membrane bound antibodies on its surfaces and which can recognize soluble or particulate antigen, but T cell receptors can't recognize soluble or particulate antigen. It can recognize only processed pieces of antigen. That antigen should be bound to MHC molecule. That antigen plus MHC molecule only can recognized by T cell receptors. Okay, so T cell receptors recognize antigen which should be bound with MHC molecule. Okay, let's see about what is MHC molecule first, then how these T cell receptors recognize the antigen. MHC molecules are termed as major histocompatibility complex molecules. Okay, so these are genetically diverse glycoproteins. So MHC molecules are glycoproteins which found on cell membranes. Okay, then there are two types of MHC molecules. The two types are class 1 MHC molecule and class 2 MHC molecule. Okay, these class 1 MHC molecules are expressed by nearly all nucleated cells of vertebrate species. Okay, nucleated cells means these cells possess nucleus in it. Okay, so in humans almost all cells possess nucleus except RBC and platelets. Okay, so almost all the cells express class 1 MHC molecule except RBC, that is red blood cells, and platelets. Next one is class 2 MHC molecule. These MHC molecules are expressed by professional antigen presenting cells and a few other cell types during inflammation. Okay, example for professional antigen presenting cells are dendritic cells, macrophages, and B cells. So this MHC molecule will form a complex with antigen and that allows to decorate their surfaces with foreign proteins or self proteins and expose this antigen to the T cells. This is a function of MHC molecule. Look into this picture. As we know that B cells are antigen presenting cells, so this B cell expresses class 2 MHC molecule. Okay, so this class 2 MHC molecule bind with antigen. Okay, this is your antigen. As we know that B cell is an example for antigen presenting cell, so this B cell expresses class 2 MHC molecule on its surface. Okay, so this class 2 MHC molecule present antigen. Okay, so this small dot, red color dot, indicates antigen. Okay, it is processed by this B cell. Okay, so this B cell has a receptor and it bind with this antigen and it processed this antigen and it present this antigen with class 2 MHC molecule to T cell. Okay, later in this video we will see in detail about the functions of T cell. Come to T lymphocytes. We can classify T lymphocytes into two major cell types. Okay, one is T helper, that is Th cells, then T cytotoxic, TC cells. Okay, these Th and TC cells can be distinguished from one another by the presence of CD4 or CD8 membrane glycoproteins which is present on their surfaces. So let's see the difference between this Th cell and TC cell. So T helper cells displays CD4 glycoprotein on its surface and cytotoxic TC cells displays CD8 on its surface. So based on this CD4 and CD8 presence, we can differentiate T helper and T cytotoxic cells. Okay, then this T helper cells recognizes antigen in complex with MHC class 2. Cytotoxic TC cells recognize antigen which is in complex with MHC class 1 molecule. Okay, so this is very important. T helper cells display CD4 protein on its surface and it recognize antigen in complex with MHC class 2 molecule. Then TC cells has CD8 glycoprotein on its surface and it recognize antigen in complex with MHC class 1 molecule. Okay, this MHC class 1 molecule is expressed by nearly all nucleated cells. This MHC class 2 molecules are expressed by professional antigen presenting cells. Okay, the ratio of presence of CD4 plus to CD8 plus means 8 plus is 2:1 ratio. So here plus means this T cells has CD4 marker on its surface. If a change in this ratio, okay, is often an indicator of immunodeficiency disease. Okay, example HIV. It is an indicator of autoimmune diseases and other some disorders. Okay, then next let's see about cytotoxic T cells. So as we know that cytotoxic T cells express CD8 glycoprotein on its surface. Okay. So when T cell receptors on naive CD8 plus T cells bind to MHC class 1 peptide complex. Okay, that is this CD8 plus T cell is going to bind with antigen which is in complex with MHC class 1 molecule. So then they become activated, proliferate and differentiate into an effector cell. Okay, this effector cell is known as cytotoxic T lymphocytes. Okay, so once the TC cell recognize the antigen which is in complex with MHC class 1, then it gets activated, proliferate and differentiated into an effector cell. Okay, this effector cell is known as cytotoxic T lymphocytes, that is CTL. Okay, next the function of this cytotoxic T lymphocytes is monitoring the cells of the body. Okay, then it will eliminate any cells that display foreign antigen complexed with class 1 MHC such as virus infected cells, tumor cells and cells of foreign tissue graft. Okay, next let's see about CD4 plus T cells. Okay, so that is T helper cells. Okay, so when T cell receptors present on the naive T helper cells, that is CD4 plus T cells, which bind to the peptide in complex with class 2 MHC molecule. Then they become activated and proliferate and differentiate into one of a variety of effector T cell subsets. Okay, there are 5 types of T helper cell. So TH cell subsets are T helper type 1 cells, TH2 that is T helper type 2 cells, then TH17 that is T helper type 17 cells, then TFH that is T follicular helper cells and TREG regulatory T cells. Okay, next let's see the functions of these TH cell subsets. So the first type is TH1 that is T helper type 1 cells which regulate the immune response to intracellular pathogens. Okay, then T helper type 2 cells regulate the response to many extracellular pathogens. So immune response to intracellular pathogen is regulated by T helper type 1 cells. Then immune response to extracellular pathogens are regulated by T helper type 2 cells. Okay, so T helper type 17. Okay, so this T helper 17 cells are so named because they secrete interleukin 7. Okay, IL-7 means interleukin 7, so it secretes interleukin 7, that's why it named as type 17. Okay, then it plays an important role in cell mediated immunity. Okay, and it may help the defense against fungi. Then follicular helper cells, which plays an important role in regulate B cell development, that is it has an important role in humoral immunity. Then regulatory T cells helps to keep the immune system in balance, that is it preventing the immune system from being overactive or underactive. Okay, it helps the immune system which doesn't attack the body's own tissues and cells, self-tolerance. Okay, thereby it prevents the autoimmune diseases. Natural killer cells constitute 5 to 10% of lymphocytes in human peripheral blood. Okay, then they are distinguished from other cells by a surface marker which is known as NK1.1. Okay, as well as the presence of cytotoxic granules. Okay, so this natural killer cells also known as large granular lymphocytes because of their appearance under microscope. Then these natural killer cells do not express antigen specific receptors. Okay, they express receptors but they won't express antigen specific receptors like T and B cells. So these are considered as part of the innate immune system. Innate immune systems or non-specific immune system. So NK cells are considered as part of the innate immune system. Receptors present on T and B cells recognize a specific antigen, so they used to give specific immune response, and that specific immune responses are known as adaptive immune response. Okay, so innate immune responses are non-specific and adaptive immune responses are specific. Next, let's see functions of natural killer cells. So natural killer cells have receptors for MHC class 1. Okay, so these NK cells express a variety of receptors for self MHC class 1. By recognizing the absence of class 1 MHC molecule, which is expressed by almost all normal cells, as we already seen in types of MHC molecule, all nucleated cells express MHC class 1 molecule. Okay, so NK cells can recognize MHC class 1 molecule, so thereby NK cell distinguish infected cells, that is the cells that should be killed from normal cells. Okay, normal cells used to express MHC class 1 but not in infected cells. So these NK cells recognize this class 1 MHC molecule and it distinguishes the normal cell and infected cells. Once it recognizes the abnormal cells by the presence of MHC class 1, it kills that abnormal target cells by releasing its cytolytic granules. So these abnormal cells including some tumor cells and some cells infected with virus. So the natural killer cells are also known as efficient cell killers. Then natural killer cells also expresses receptor for immunoglobulin, that is for antibody molecules. So these receptors bind with antibodies that bind with the protein from the pathogen on the surface of infected cells. Okay, thereby these natural killer cells connect with a variety of abnormal cells. So once the antibodies brings the natural killer cells in contact with the target cells, that is abnormal cells, then this natural killer cells releases its granules and induces cell death. Okay, and also NK cells are powerful producers of chemokines and cytokines, particularly interferon gamma. So this interferon gamma is a cytokine. It has antiviral, immunoregulatory, and anti-tumoral properties. Okay, it responds during the early stages of infection before the adaptive immune responses develop. So natural killer cells gives an immediate immune response. In the next video, we will see in detail about cells arises from the myeloid progenitor. Okay, thank you for watching my videos. If you like my video, please do like, share and subscribe.